Hi everyone, my name is Tabea Soelter, and today I am talking to you guys about my poster titled The Role of Cell-to-Cell Communication in Alzheimer's Disease. And as a neurodegenerative disease, one of the primary hallmarks of the disease is neuronal loss, but it is known that non-neuronal cell populations maintain brain homeostasis and neuronal health. Non-neuronal cell populations are comprised of microglia, such as macrophages, which are usually involved in neuroinflammation, and macroglia, for example, astrocytes and oligodendrocytes. Astrocytes provide metabolic and nutritional support to neurons, while oligodendrocytes are responsible for the myelination of the axons of neurons, which usually facilitates neuron-to-neuron interactions. Neuron glia and glial cell crosstalk is via chemical messengers and enables normal cognitive function as neuronal health and functionality is maintained. Reactive microglia have been previously implicated in the activation of reactive astrocytes, though, which are known to be neurotoxic and lead to neuronal degeneration throughout disease progression. Yet the causal mechanisms of altered neuron glia interactions underlying diseases like Alzheimer's disease are not really fully understood. And so single nucleus RNA sequencing has the ability to address cell type specific gene expression changes and allows the application of in silico methodologies that can predict ligand receptor interactions and infer cell-to-cell communication. And some methods like NicheNet can even provide predicted target genes that are affected by such ligand receptor pairs. And so in this particular project, we use publicly available post-mortem human avian control brain single nucleus RNA sequencing data. And we identified ligand and target genes in both AD and control neurons. And we looked at the cell type specific expression of ligands across the sender cell populations, in this case in astrocytes and in oligodendrocytes. And so for this study, we downloaded the public AD single nucleus RNA sequencing data, which, as mentioned previously, is post-mortem human hippocampus. And we have 9 AD samples and 8 controls. And so this particular data was then aligned and quality controlled using the 10x Genomics Cell Ranger pipeline. And then after that, we performed pre-processing using Seurat and R&R Studio, including the integration of both of our conditions, so AD and control. And so once we integrated our data and integrated both the conditions, we actually then clustered our nuclei at a 0.2 resolution, which would determined using clustery. And then we assigned the cell types based on differential gene expression and known marker genes from the literature and the database called CellMarker, which you can see below. And so then in order to identify the cell-to-cell interactions that are different between our AD samples and our control samples, we applied a differential NicheNet analysis for astrocytes and oligodendrocytes as our sender cell types and neurons as our receiver cell types. And so overall, we were able to identify differentially expressed ligands and target genes in both astrocyte neuron and oligodendrocyte neuron interactions. And so we are able to observe some overlap between the different sender cell types, but we also see differences between these cell types, which some of them I'd like to highlight. And so in figure 1, we see, interestingly, the ligand AGT, which is highlighted in the purple star, which has relatively or overall low ligand activity, but it actually regulates the differential expression of many genes in the AD neurons, including PTGDS. Which actually encodes a prostaglandin synthase, which is a neuromodulator, which affects neuron-to-neuron communication. And so PTGDS is the same gene affected by a ligand identified in our oligodendrocyte neuron interactions, which is POMC highlighted with a black star in figure 3. And so both oligodendrocytes and astrocytes have different ligands that affect the same target genes, PTGDS, which is quite interesting. However, when we're looking at predicted differentially expressed target genes in the astrocyte neuron interactions in our control samples, we actually see the ligand TGFB1, which is highlighted in a peach star in figure 2 in our control astrocyte neuron interactions, which has high regulatory potential, which are involved in cell-to-cell adhesion and inflammatory response and in the regulation of apoptosis. And then in our control oligodendrocyte and neuron interactions, we again see some overlap between our astrocyte neuron interactions and our oligodendrocyte neuron interactions in our control. So we again see the same ligand, the TGFB1 ligand here as well, but we see that this particular ligand in the oligodendrocytes drives most of the differentially expressed genes here. So overall, we were able to identify differentially expressed ligands and target genes in both the astrocyte neuron interactions and the oligodendrocyte interactions. And so while we do see some overlap here, we also see significant differences. And so we were able to identify ligands that are either cell type specific, and some of them are shared between astrocytes and oligodendrocytes. And so for the future, we would like to evaluate the overlap of the ligand receptor pairs between these different groups, so between the astrocyte neurons and the oligodendrocyte neuron interactions, to further investigate and understand why there is this overlap and what exactly that means for Alzheimer's disease. We also would like to analyze cell-to-cell communication between other cell types. So for example, we have the oligodendrocyte progenitor cells or microglia, and we'd like to look at these interactions between neurons as well. And ultimately, we'd also like to do the analysis with neurons as the sender cell types and oligodendrocytes and astrocytes as the receiver cell types. So overall, we do see differences in cell-to-cell communication between the conditions and cell types, and identify target genes that are associated with neuron-neuron communication, inflammation, cell survival, and neuronal excitability. And with that, I'd like to thank my lab, and I thank you for stopping by my poster. And if you have any questions, please submit them or reach out to me. Thank you.